Welcome to Roadmap to Residency Evidence-Based Application. This video is summary of all the videos that we have talked about. And in this video, we will be mainly focusing on the microbial diagnostic, how to identify bacteria. We have talked about in detail the identification of bacteria in our previous videos. So this is will be just a summary and some important points. So identification measures of bacteria include microscopy, which is staining, growth on enrichment, selective differential or characteristic media, biochemical test, immunological techniques, or molecular methods. After the bacterium is identified from specific clinical samples, it is used in susceptibility tests to find which method of control is most effective. Staining reactions. The presence of certain structures and staining reactions aids in their identification and classification to render microscopic and semi-transparent objects visible, to reveal their shape and size, to produce specific physical or chemical reactions. So simple staining brings out the morphology the best. Differential and special stains are necessary to bring out characteristics like flagella, capsules, spores, and metachromatic granules. Gram stain divides bacteria into gram positive and gram negative. G. Nielsen stain divides them into acid fast and non-acid fast. And fluorescent dyes brings out special characteristics and fluorescent antibody technique, which enables to identify them. Cultural characteristics provides information for the identification of the bacterium, and these characters revealed in different types of media are noted. Now, while studying colonies on solid media, following characteristics are observed, like size, shape, margin, surface, the elevations, age, color, structure, consistency. Now, in fluid medium, following characteristics are observed, like the degree of growth, whether there is absence, scanty, moderate, or abundant growth, presence of turbidity and its nature, presence of deposit and its character, nature of surface growth, ease of disintegration, and order. Resistance and other aspects. The resistance of the organism is tested against number of parameters which helps differentiation and identification of the organism, like heat, low concentration of disinfectants, antibiotics, chemotherapeutic agents, bacteriocins, and etc. sort of aspects. Now, other aspects studied to classify and differentiate the species may be requirement of oxygen, the need for CO2, capacity to form pigments, and power of hemolysis. Some of the biochemical properties are very important. That is the test for metabolism of carbohydrates and related compounds, test for metabolism of proteins and amino acids, test for metabolism of lipid, test for enzymes, and some combined test. Test for metabolism of lipids, which may be hydrolysis of tributylene, which is an emulsion of micro droplets of the fat. Tributylene in a solid medium makes it opaque. Certain lipolytic organisms remove the opacity by converting the fat into water. Test for enzymes like catalase, oxidase, and urease tests are very important. Catalase tests demonstrate the presence of catalase, an enzyme that catalyzes the release of oxygen from hydrogen peroxide. Oxidase test depends on the presence in bacteria of certain oxidases that will catalyze the transport of electrons between electron donors in the bacteria and a redox tie. Urease test. Bacteria, particularly those growing naturally in an environment exposed to urine, may decompose urea by means of the enzyme urease. Some of the immunological techniques, like the culturing of certain viruses, bacteria, and fungi, and parasites from clinical specimens, may not be possible because the methodology remains undeveloped. For example, in case of Treponema pallidum, hepatitis A, B, C, and Epstein-Barr virus, unsafe, like rickettsial infections, impractical for all but a few clinical microbiological laboratories like mycobacteria. Cultures may be negative because of prior antimicrobial therapy. Now, under these circumstances, detection of antibodies or antigens may be quite valuable diagnostically, as immunologic systems are easy to use, give relatively rapid reaction endpoints, and sensitive and specific. Now, this is a picture from your first aid, very important. Please remember this by heart. So when we have the gram-positive organism, we may have bacilli, we may have a cocci, or we may have a branching filaments. Whenever we have a branching filaments, it may be aerobic or anaerobic. If it is aerobic, it is nocardia, and if it is anaerobic, it is actinomyces. Talking about bacilli, it again may be aerobic or anaerobic. Aerobic organisms include Listeria, Bacillus, and Corynebacterium, and anaerobic means Clostridium and Cutibacterium. But no, if we have cocci, we have to go for catalase test. Now, if it is catalase positive and it forms cluster, then it is Staphylococcus. Now, again in Staphylococcus, we have to differentiate which sort of Staphylococcus. For that, we have to do coagulase test. If the coagulase test is positive, so Staphylococcus aureus have coagulase plus catalase positive, Whereas if coagulostase is negative, then we have again 2. Then we go for novobiocin sensitivity. So if there is novobiocin sensitivity positive, it is staph epidermidis. If not, it is staph subtropiticus. Very, very important, this pathway. Again, if it is catalysis negative, we have streptococcus. Then we go for hemolysis reaction, alpha, beta, and gamma hemolysis. Gamma hemolysis means there is no hemolysis, and beta means there is complete, whereas alpha means there is partial hemolysis. If there is alpha hemolysis, we go for optogen sensitivity and bile solubility. If it is negative, then they are very dense group of streptococci. If not, then there are streptococcus pneumoniae. Whenever there is beta hemolysis, we go for bacitracin sensitivity, that is group B, streptococcus agalacticae. Or if bacitracin sensitivity is positive, then it is group A, that is strep pyogenes. If there is no hemolysis, then we see the both in 6.5% sodium chloride, and we check the pyrrolidonyl aminopeptidase, PYR status. If it is negative, it is strep gallolyticus. If it is positive, it is enterococcus. So this chart you have to go on time to time. Can remember this by heart, because this will help you answer many questions in the real examination. Again, if you have gram-negative organism, it may be cod rod, it may be Coccobacilli, or it may be Diplococci. If it is Diplococci, it is aerobic. Then we go for maltose fermentation. If it ferments the maltose, then it is Neisseria meningitidis. If not, it is Neisseria convea. If it is Coccobacilli, we have straightforward Haemophilus influenzae or Bordetella pertussis. If we have cod rod, then we go for oxidase test. Then these are generally oxidase positive. Then we go with a specific character, like if it is grows in 42 degrees Celsius, it is Campylobacter jejuni. If it grows in alkaline media, it is Vibrio cholerae, and if it produces urease, it is Aspergillus. Or it can be gram-negative bacilli too, which generally ferments lactose. If it ferments lactose, then we see if it is a fast fermenter or a slow fermenter. If it is fast fermenter, it is E. coli and Klebsiella. If slow, it is Citrobacter. Bacilli lactose fermentation is negative, then we go for oxidase test. Now, oxidase positive is Pseudomonas, oxidase negative is H2S production. If there is oxidase negative, we go for H2S production test. If it is also negative, this is Shigella and Yersinia. If it is positive, it is Salmonella. Now, you may find this very difficult, but the only key to remember this is to go step by step and remember this, review these charts every single day while you encounter different questions and while you go through UWorld and all. So please remember this by heart. Thank you for watching.